and welcome to episode 18. Today we're going to be discussing gene modification. In this series on genetics, we started with Mendel's first experiments into the field, and then the following discoveries. We covered meiosis and transcription and translation, DNA synthesis and gene expression. Today's episode is about gene modification, and it'll be the last episode in this series. This episode is unique in that it's the first episode that will discuss man-made breakthroughs in genomics and how they affect the world we live in today. Biotechnology is an emerging field with tremendous medical potential, and artificial gene modification is only a small aspect of it. As I'll cover in this podcast, the ability to modify and manipulate genes on a molecular level has revolutionized our world, so much so that it affects the food we eat, our medicine, and how we understand the history of life on Earth. This is actually a good place to start our episode, as the history of life has largely been revealed by the study of the change of genes over time. Single-celled organisms like bacteria and archaea have relatively simple genomes. The larger the organism, the larger the genome. Single-celled organisms of all sizes can interact with one another through a process called lateral gene transfer. If you understand reproduction to be vertical gene transfer, like genes being passed down a family tree, then what would lateral gene transfer be? As you might have guessed, it's the transfer of genes between two independent, mature cells. Archaea and bacteria keep their genomes in the form of a giant loop, along with smaller loops of DNA called plasmids. These plasmids, as well as replicated chunks of the larger chromosome, can be traded between cells, where they get incorporated into the receiving cell's genome. Evidence shows that almost all bacterial and archaeal species are affected to some degree by lateral gene transfer. And this has prompted some scientists to question the current understanding of the evolutionary branches of archaea and bacteria. Eukaryotes like plants and animals and people also have natural techniques for gene modification. All organisms, including bacteria and archaea, have things called transposable elements. These are small little snips and tidbits of DNA, short, tiny little sequences that can be inserted into almost any other location in the cell's genome. These elements are copied, and the copies can be stitched into a new DNA sequence, causing negative, neutral, or positive mutations. Transposable elements are kind of like a boggle in the cell. You shake it up and you see what you get. Humans have numerous transposable elements, but many of these genes produce inactive proteins, as we lost the genes for an associated activator or something like that. So we produce a bunch of transposable elements, but we don't have the blueprints for the proteins that activate them. So the elements just float around in our cells, doing nothing, until they get broken down and their molecules recycled. In case you are wondering, yes, these transposable elements are a part of the bulk of so-called junk DNA that litters a eukaryotic genome. The eukaryotic cell's genome also carries with it strange little repeating sequences. 2 to 6 bases makes a microsatellite, or STR for short tandem repeats. Well, anything longer than 6 bases is a mini-satellite, or a VNTR, for variable number tandem repeats. Okay, so what are these things? Why do they matter? They're sequences of DNA with lots of particular bases in an alternating repetition, AC, 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 or TG, TG, TG. If you consider the random DNA sequences to be kind of like a key in a lock mechanism, a key requires, an appropriate lock, or vice versa. In the DNA, this means that the sequence can only bind to a complementary strand of DNA. During meiosis, the homologous chromosomes align next to one another, with similar DNA sequences associating in a way that makes the homologs line up nicely. These tandem repeating sequences, however, can mess up this nice arrangement. Because the sequences are so similar, the homologous chromosomes can accidentally get them mismatched, which makes them slightly out of alignment. During recombination in later stages of meiosis, these misalignments lead to the tandem sequences getting unequally distributed between daughter cells. Some daughter cells have more tandem sequences or longer sequences, while others have the opposite. Now this is where stuff gets really interesting, because this means that every person has a unique frequency of tandem sequences in their genome. This is like a DNA fingerprint, and it's the characteristic used in forensics to study DNA evidence. The criminal justice systems uses the evidence gathered from DNA fingerprints to determine innocence or guilt, to exonerate those falsely imprisoned, or to identify individuals engaging in repeat offenses. You might be left wondering, well, what about the misaligned chromosomes? Doesn't that lead to cells having differently sized chromosomes, even within the same organism? And the answer is yeah, it does. And the cell's response to this problem is a major source of genetic diversity in eukaryotic organisms. When the misalignment occurs in meiosis, gene-sized segments, basically long sequences of DNA or small chunks of the chromosome, can be deleted or added. And when the cell goes through this DNA to check for errors, it notices the misalignment and it produces a copy of a nearby gene to fill the gap. The end result is gene duplication, or two copies of the same gene right next to one another on the chromosome. This is convenient, because if the cell needs to express these genes, a regulator complex only needs to make one pass to transcribe two copies of the protein. It's basically twice the production for the effort of one. This has the potential to make the cell respond more strongly to the associated stimulus. For a simplified example, consider a drop in pH. If the drop is strong enough, this stimulus causes a cell to express proteins that protect it from acidic conditions. The cell with two copies of this gene would produce twice as many protective proteins as a cell with just one copy, and can thus protect itself better from the acidic conditions. The better protected cell is more likely to survive and reproduce in the future, pass on its two copies of the gene. Furthermore, with two copies, the cell can survive if one of the copies mutates, as it still has one functional copy of the gene. This is also where more genetic diversity comes in, because if you have a duplicated gene with a mutation, that mutation can induce a negative, neutral, or positive effect. Most of the time, it'll be a negative or a neutral effect by producing the non-functional sequences called pseudogenes. But on rare occasions, the mutation will have a positive effect. The mutation might improve the protein's efficiency or effectiveness, or it will slightly alter it in a way that it can associate with the unaltered proteins to create a complex. As both gene copies get expressed at once, their products would easily be able to associate and form a functional protein complex, or an enzyme activator relationship, or something like that. In this way, groups of related genes stay together in a kind of form-follows-function relationship. This group of genes is called a gene family, and this operates in much the same way as a taxonomic family does, in the study of evolution. Gene duplicates create gene families, and the slow accumulation of mutations in these duplicates and in these gene families can help diagram when related species diverged from a common ancestor. I'm going to transition now to briefly talk about the scientific fields that are focused on studying genes and gene expression, and the equipment and techniques they use to acquire their data. Perhaps the most general field that I want to discuss is called functional genomics. Those who work in the field of functional genomics dedicate their work to studying the complex relationships between regulatory molecules, genes, and the proteins they express. They seek to understand why genes are grouped or activated in the ways that they are, and how the expression of genes is coordinated in response to an outside stimulus, or as part of the organism's natural development and maturation processes. Another field of genetics is proteomics, or the study of proteins. Where the genome is the organism's full set of genes, the proteome is the organism's full set of proteins. Proteomics studies the interactions of many or all of the proteins in an organism's body, all operating at once to maintain homeostasis to keep the organism alive. Both proteomics and functional genomics use a powerful tool called a microarray. This is basically a flat piece of silicon substrate, and it's studded with millions of little biomolecules like proteins or single-stranded DNA sequences. The biomolecules studded into the substrate are known. Scientists know what they are. So they know that when they introduce foreign biomolecules to the microarray, they can see which of the known molecules will react or bind to the unknown foreign molecules. In this way, the microarray is extremely useful for seeing how genes are expressed at different ages or in response to outside stimuli, and how proteins interact with one another inside the cell. Understanding how individual systems interact to sustain a greater whole is the whole purpose of systems biology. The systems biologist studies the emergent properties of massively complex chemical systems, properties like metabolism or growth. The systems biologist seeks to unify the knowledge of functional genomics and proteomics into a cohesive whole, which would then describe the macroscopic effects of uncountable chemical reactions in trillions of cells to maintain the life of a single organism. In order to do this kind of research, scientists need more than just the humble microarray. They need other equipment, more sophisticated equipment or stuff that has different abilities. Stuff like a Pcr machine, a Deoxy sequencer, Crispr, or some other recombinant DNA technology. A PCR machine is a device that allows scientists to quickly print out millions of copies of a specific DNA sequence. The known DNA sequence is put into a watery solution, where the temperature is increased until the hydrogen bonds in the DNA strings actually break apart and the strands separate. DNA synthesis is then initiated through primers, which are, little RNA protein bits, and the associated primers of the known DNA sequence are mixed into the solution. And so the temperature is lowered, and the primers can bind to the single-stranded DNA. This attracts the polymerase molecules. They're also thrown in the solution too. And these synthesize a complementary strand of DNA onto each single strand, starting from the primers. And so the original strand of DNA has now been doubled. And so then another cycle of heating happens so that they break apart. And it cools again, so that the primers attach and the DNA polymerase makes more strands. And so each sequence doubles. Another cycle of heating and cooling will double the DNA again. Another cycle, another doubling. The process is exponential, so it only takes 20 to 30 cycles to produce millions of copies of your desired DNA sequence. This technique allows us to take small, tattered fragments of DNA found in preserved tissues or fossils from extinct species and amplify them a millionfold. Then it becomes easy to isolate the sequence and study it to see how it fits into the ancestral lineage of currently living species. This is how we learned about the interbreeding between Neanderthals and various groups of Homo sapiens. It's how small DNA fingerprints can be amplified and examined for forensics for criminal justice. So once the PCR machine gives you 1 billion copies of your desired sequence, the next thing you want to do is figure out what your DNA sequence actually is. For this, scientists use a process called Deoxy Sequencing, or Sanger sequencing, after the scientists who devised the method. First, a solution is prepared for DNA synthesis. This means it's rich in deoxyribonucleotide bases, the A, T, C, and G, all ready to be incorporated into a DNA strand. But it also includes biomolecules called dideoxyribonucleic acids, which lack the critical OH group necessary for DNA synthesis. Recall that DNA polymerase can only add new nucleotide bases to the 3' end of a growing DNA strand, and it does this by interacting with the OH group on the 3' carbon. But if there's no OH group there, there's nothing for DNA polymerase to interact with, and so it can't add another base. Because of this, whenever a dideoxyribonucleotide is added to a strand of DNA currently under construction, the DNA synthesis terminates. The enzymes can't add another nucleotide base onto the 3' carbon's OH group, because it just isn't there. So when you have proteins synthesizing copies of a particular DNA sequence in this solution, you end up with a large number of copies of all kinds of different lengths. Each copy was built up using regular deoxyribonucleotides until a dideoxyribonucleotide was added, and this terminated DNA synthesis. The key thing to understand here is that this means that the dideoxyribonucleotides expose which nucleotide base sits at which position in the DNA sequence. When you line up the DNA copies side by side according to their length, from smallest to largest, the dideoxyribonucleotides on the ends tell you what base exists at that point in the sequence. You can literally go from your smallest copy, say 3 bases long, and see the end nucleotide by studying its spectroscopic signature or its light signature. This tells you the nucleotide at position 3. The next copy, 1 nucleotide base longer, tells you the nucleotide at sequence 4. The next one will tell you it at sequence 5, and then 6, and so on. Sanger sequencing was one of the first techniques developed for DNA sequencing, so it's relatively labor-intensive and time-consuming. Newer sequencing technologies have been developed that increase speed and accuracy. PCR techniques can be used to amplify hundreds of different genes at once, and these can all undergo simultaneous sequencing in an effort called a massively parallel approach. Other techniques operate on the molecular level and involve the literal pulling of the single-stranded DNA through a pore. The pore's shape is altered in a certain way depending on the nucleotide base currently passing through it, and the sequence of responding pore shapes can be used to determine the DNA sequence itself. Other recombinant DNA techniques use various methods to identify a specific DNA sequence, and either remove it, copy it, or insert it into a different gene or genome. These isolated DNA sequences are packaged with proteins or inserted into a small plasmid for transport, where they can be literally injected into host cells and incorporated into their genome. A growth hormone gene was isolated and used in this way as a medicine for treating pituitary dwarfism. And it turns out that it can even be used cosmetically to increase your height, or improve your bone density and muscle mass. This takes me to an exciting subject called CRISPR, or CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats. Basically, a CRISPR is a sequence of prokaryotic DNA that can identify and cut out DNA that doesn't belong to the organism, like the DNA injected by a virus, or a deleterious gene handed over during lateral gene transfer. The CRISPR molecule forms a complex with a protein called Cas, and together they form a functional gene editing group that protects the prokaryote by removing bad DNA. Scientists were able to modify the Cas protein into something called Cas9, and this CRISPR-Cas9 complex can be used to modify genomes at will. Basically, the CRISPR-Cas9 complex needs only be guided by a particular RNA, and it can cut out any part of the genome you'd like. The sheer versatility and utility of this feature is indescribably exciting in the biological fields, especially in those that deal with treating genetic diseases. Gene therapy is a tricky science. Once an individual has identified a problem gene within their genome, like the genes that cause Huntington's disease or cystic fibrosis, gene therapy seeks to counteract the damaging genes with healthy genes. The trick to this science involves figuring out a way to get healthy alleles of a gene or genes into cells with the unhealthy alleles. This must be done such that only the desired tissues are affected. It must be done with the right concentration and dose, at the right time in the organism's development. Furthermore, the gene therapy must find a way to silence the unhealthy alleles, so that only the healthy alleles are expressed. Problems with any of these variables can cause a host of potentially serious, sometimes fatal complications. Genes can be inserted into cells through mechanisms that work like a virus, injecting the DNA and a minimal host of regulatory proteins into a cell such that the host enzymes incorporate it and begin expressing it. These techniques often involve the use of retroviruses, which catalyze the insertion of the viral DNA into the chromosomes of the host cell. Gene therapy is used to treat a disease called SCID, or Severe Combined Immunodeficiency. Basically, a gene on the X chromosome that's mutated such that it no longer works, and this effectively neuters the individual's immune system. The gene codes for a receptor on the T cell, which is an important cell in the immune system, and when that gene gets mutated, it renders it non-functional. The individual has non-functional T cells, so even a minor infection can be potentially fatal. For the longest time, treatment was limited to keeping these kids in a sterile room or in a bubble for their entire lives. To address this issue, researchers took 10 male infants with SCID and extracted the stem cells from their bone marrow that code for T cells. A retrovirus was used to put a healthy, functional version of the gene into the genome of the stem cells, and the stem cells were then placed back in their infant hosts. Of the 10 infants with SCID who underwent this gene therapy, 5 went on to grow up with normal T-cell behavior and normal immune function, having been effectively cured of SCID. 4 of the children would develop cancers due to uncontrolled T-cell production, one of whom would die from it. The 10th child failed to respond to the initial treatment, but he was treated with donor cells and has since lived a normal life. As you can see from this example, gene therapy is not a clean-cut science. What works for many may not work for some, and may even lead to fatal complications in others. The technological sophistication of our gene therapies has to be improved to minimize dangerous errors like the ones that cause cancers later in life. We need to think about how gene therapies can be used, not only to treat, but to permanently cure and put an end to genetic diseases that have plagued families for generations. This opens the door for cosmetic and elective gene modification, but I'll get to that at the end of the episode. It goes without saying that gene modification isn't limited to medicines. If you eat food, chances are you've eaten genetically modified food, or food produced by a genetically modified organism, also called a GMO. I don't mean organisms modified slowly through artificial selection over thousands of years. I'm talking about organisms who've had their genes radically modified overnight in labs by people sitting hunched over their desks looking into microscopes. This kind of gene modification has revolutionized our world by providing food that lasts longer, handles temperature extremes much better, and grows larger and brighter with better yields, and some would say a better taste, although that's kind of a matter of personal opinion. Probably the most famous GMO is called golden rice. Let me create the context for you. Half the world Half the world eats rice as a staple food, as an integral part of their diet. However, rice doesn't provide the full complement of nutrients that human bodies need. For example, rice doesn't have vitamin A. Its leaf and stem tissue produce a protein called beta-carotene, and this gets modified into vitamin A in the human body. But the actual rice that you eat, the actual carbohydrate-rich, little rice grain, doesn't have beta-carotene. You can't derive any vitamin A from the rice. This can be problematic, as vitamin A deficiency can lead to blindness. And tragically, nearly 250,000 children in Southeast Asia develop blindness every year because of a vitamin A deficient diet. This is clearly a problem. So what's the solution? Scientists wanted to see if they could get beta-carotene to be expressed in the rice tissue itself. So first, they identified a protein in the rice that can be modified into beta-carotene with the help of just 3 enzymes. So they pretty much found a similar protein in the rice, it's kind of like beta-carotene, and they found out that with just 3 different enzymes, each one coming in and making a minor modification, they can turn that molecule into beta-carotene. The genes that code for these enzymes were isolated and then strung together with endosperm regulatory sequences. These would ensure that the genes are expressed in the endosperm, which is the fancy word for the tissue that eventually becomes the grains of rice. The researchers then identified a bacterium with a plasmid capable of injecting its own DNA sequences into the DNA of a host plant. They used this bacterial plasmid as a vehicle to deliver the enzyme genes into a rice embryo. As the embryo grew into a plant and produced rice, the researchers found that much of the rice was a rich yellow color. As the rice endosperm grew, the endosperm regulatory sequences made sure the genes were activated. This produced the enzymes necessary to turn the natural precursor molecule into beta-carotene. The product rice was packed with beta-carotene, which gave them their rich yellow color, and also their name, golden rice. As the human body makes vitamin A out of beta-carotene, the golden rice effectively solves the vitamin A deficiency problem. But despite these apparent benefits, golden rice hasn't been approved for commercial production and sale in any country. However, rice is hardly the only genetically modified plant. Potatoes, tobacco, alfalfa, maize, papaya, corn, cotton, tomatoes, canola, eggplant, apples, bananas, squash, soybeans, and carrots are just a handful of the total number of modified plant species. When scientists perform some kind of genetic modification, they typically try and make the species more durable and efficient from an agricultural context. This means they find ways to repel insects and other herbivores that would damage the crops, ways of minimizing the impact of weeds, and improving the nutritional content of the produced fruit, as well as the crop yield, so the total fruit that gets produced per crop. For example, the first GMO crop approved for sale in America was a strain of tomatoes given the name Flavor Saver. The Flavor Saver tomato had a longer shelf life, as its engineers were able to disable the expression of an enzyme that eats away structural proteins in the cell walls. Ordinarily, this gene would cause the tomato to get soft and mushy as it aged, but its suppression allowed the tomato to stay firm and edible for significantly longer periods of time. This also makes it easier to ship tomatoes, as they last longer in transit. Another GMO crop is called the Arctic apple. These apples were modified to silence a gene expressing polyphenol oxidase, which is responsible for the browning of fruit tissue exposed to the air. As a result, Arctic apples take significantly longer to brown from oxygen exposure compared to other apples. Cavendish bananas are modified to express nearly 10 times as much vitamin A as unmodified bananas. Drought-guard maize is a species of corn that can endure prolonged periods with little to no water. Other modifications can make the plants tolerant to various herbicides or insecticides. If the crop can be made immune to a particularly strong herbicide, then that herbicide can be used to kill weeds without harming the crop itself. Similarly, powerful insecticides can be used to kill parasites and insectoid herbivores without harming the crop plants. Crops like potatoes, squash, zucchini, and cucumber have all been modified to be resistant to various viruses. Plants aren't the only genetically modified organisms. Many animal species have also been modified to suit our agricultural needs. Researchers in China, Argentina, and New Zealand have all independently engineered genetically modified cows that express human proteins in their milk. These cows have been effectively engineered to produce human breast milk instead of natural cow milk. A lineage of pigs called enviropigs were engineered to break down phosphorus compounds in their food, leading to a huge reduction in the amount of phosphorus they excreted. This is really important for ecosystem health, as the waste from thousands of pigs all shitting in the mud on an industrial agriculture farm creates runoff that's rich in phosphorus. When this waste phosphorus gets into water sources, it acts as a food for all sorts of algae species. When the algae eats this phosphorus, they also end up consuming a lot of oxygen, which can create dead zones in the water where there's no oxygen for fish. So by modifying pigs to reduce the amount of phosphorus they excrete, we can minimize algal blooms in nearby bodies of water and better protect fish populations. Even fish themselves are being modified. There's a company called Aqua Bounty that's patented a GMO salmon that they claim can be raised to maturity in half the time as a normal salmon. Besides agriculture, animals are also modified for scientific purposes. Fruit flies, sea anemones, and zebrafish are model organisms routinely used in genetic modification experiments. There's a famous study using fruit flies, where the researchers were able to activate the genes for an eye in the cells on a leg. Basically, they were able to grow a fly's eyeball coming out of its kneecap, just by messing around with regulatory sequences. The eye on the kneecap couldn't see because none of the genes for, the proper neural structures were set up or expressed, but there was literally an eye, like a fly's eyeball, growing as like a little red nub coming out of its leg. And that's what happens when cells are told to express genes that they don't normally express. Because remember, every cell in your body has your entire genome, but because of gene regulation, they only express, a limited amount. But this just goes to show you what can happen if you start messing with that regulation. A fluorescent protein found in jellyfish has been transplanted into pigs and cats, and this literally makes some of the animal's tissue glow with like a soft fluorescent light. These fluorescent proteins are extremely useful because they can be tracked and monitored as they move throughout an organism's body. This way, they can illuminate the pathways and responses cells make during transplants or when exposed to certain diseases. Other organisms like sheep, pigs, and goats are modified so that their organs are much more acceptable to human hosts. This has an important medical purpose, as animal organs with these modifications are much less likely to be rejected by the body, and they're also much more likely to integrate fully and to operate effectively in a human body. Basically, should you get into a car accident and need an organ transplant, these genetically modified pigs have organs that your body is much more likely to accept, because it's their human organs technically, they're just grown inside of a pig. From food to medicine, genetic modification can be used to create a more sustainable world. GM crops can give more yields, grow more sustainably, and feed more people. GM animals can provide novel sources of nutrition, research opportunities, and organs better suited for transplant. With some of the technologies that I discussed, we can potentially start modifying our own genomes, although that opens the door for a very important ethical discussion. How far should we go with our own genetic modification? What's acceptable? For example, as a matter of personal freedom, should we let people modify their somatic cells as they desire? Or should we let people have the ability to modify their germ cells, which would then be passed on to their offspring? If someone modifies their germ cells, they're basically modifying the genome of any offspring they choose to have. Is it ethical to give birth to a child with a cosmetic or a elective gene alteration that the child themselves did not consent to have? In the case of a genetic disease, you might think it's unethical not to cure it. You might even argue that once made aware of their heritable genetic disease, people should be compelled to get gene modification. On the other hand, shouldn't cosmetic modifications be just as tolerated? It's fine for someone to choose to modify their genes to make their eyes blue, for example, but what if that modification is designed in such a way that it gets passed on to the person's children? You might think, well, blue eyes aren't that bad, but what if it's more extreme than that? What if the individual wants to change their skin color, their limbs, their genitals, or their facial structure on a genetic level? People already do this with piercings and tattoos and body modification, so if people could do it genetically, they would. I don't see how you can say they wouldn't. People do crazy stuff. People do wild things. And so if the technology surrounding gene modification becomes more and more sophisticated, we'll have to have these conversations. We'll have to face these ethical issues, and we'll have to address how to responsibly handle our species' genome for the sake of all of our future generations, all of our future descendants. Well, I think I can end the podcast on that high note. I hope this episode helped you shine a little light into the world of genetic modification. It's an important science that's becoming increasingly common in our technology-dependent world, so it's important that you understand at least the basics, the basics of gene modification. I hope you give a little thought to ethical questions surrounding gene modification and the potential places that gene modification can take us in the future. As always, thanks for listening.
Would you like to support the BioLogic podcast? It's super easy. When you open a new episode, press the like button or share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.